afternoon. My name is André. Bruinjé, or in Portuguese it would be Bruinjé. Well, what I'm going to show you guys is the beginning of my PhD project. So I am in the Ecology and Conservation Biology graduate program in Universidade Federal do Paraná, in south of Brazil. But the majority of my work is being done in northeastern Brazil, in Universidade Federal do Rio Grande do Norte. So I'm very thankful for all the both labs and my advisor and co-advisor. So the outline of the presentation here is just talk briefly about sexual selection in relation to polymorphism, present you the model species that we used, how we assess the issue with in intra and intersexual behavioral experiments, and showing you our preliminary results and our ongoing and further steps. So usually I have to put out the framework to the audience about the sexual selection. Luckily I don't have to do it here today. But basically, in the selection in sexual context, we tend to see it as towards extreme phenotypes. And what makes us to think that the not chosen trait would be negatively selected. But therefore, how can we explain the maintenance of polymorphic trait within this framework? So that's the main question that got me into my PhD, and so we decided to assess this question using lizard species as model system. So we are using actually the striped lava lizard, Tropiduridae semitenniatus, which is a rock-dwelling species that occurs in the majority of the states in northeastern Brazil. So this species, it presents a color polymorphism within the males. The males have these two types of males: the yellow males that have these patches, yellow patches in the thighs and cloaca, and the black males. And the females don't have a patch at all. So this coloration also increased during breeding season, and they kind of have also a patch showing behavior, which I tried to put here, but this is just a picture from one of the experiments. But they have this behavior of kind of showing among more other complex behaviors, and so these all together make us think that this patch might be sex related. So we sampled 160 individuals. Actually, we sampled 240, but 160 were okay to be used in the experiments. In the same population in João Câmara, we kept them individually in the lab with fixed photoperiod, temperature, and fed ad libitum during this last breeding season that just ended. And then to assess if and how the morphs differ, we went first to explore the first differences. So actually, does this population have morphs? Because what we had before was just a description, but based in other populations, not these particular. But okay, so we found the two types of coloration in this population, and then we went to explore some morphometric measures and differences between them. So these are some of the measures that we measured, together with head size measured, and then in the plot what we can see here is that this is the SPL, the size, we can say, of the animals between sexes. So the females are significantly smaller than the males. And here, what we have is that the females are different from the males, but the males, the yellow males, are different from the black males. So the black males tend to be larger and bigger, and the yellow ones tend to be smaller. The same pattern we can see with the head size. So we went. The second thing we want to do went to throughout experimental approach within sexual selection context. So we assessed the intersexual selection throughout visual mate choice and throughout chemical mate choice experiments, and intersexual selection throughout male competition over a resource experiments. So the visual mate choice experiments, what we did is a female is put in a tank where it get to choose between a yellow male and a black male. The two males can see each other, but the female can see both of them. And what we have is, so we made observations in the place where the female is every 10 minutes for 6-hour trials. It's important to mention that the males were size paired. And altogether we ran 35 combos of a female plus a male pair. So it was 35 females and 70 males. And we used the choose beginning criteria, which is we only take into account data after the female starts to behave, or to at least after she is able to see both options. The female chemical choice experiment was pretty much the same, but as the female don't have to see the males, so we can, it's kind of easy because we can use the same males or the same male sense in multiple tanks. So what we have here is like a tank like the other one, but the female gets to choose between the yellow one, the black one, or in a neutral place. We made 10 minutes, every 10 minutes, observations every 10 minutes during 5-hour trials. And paper stripe with the scent of the males. We used 19 male pairs and 29 not previously experimented females. Totalizing, but we ran this experiment for 4 consecutive days, randomizing the females and the pairs and the place and the tanks and all that we could. It's important to mention that the males were also size matched, and in a way that we had no repetitions. And we used the same choose by beginning criteria. The resource competition experiments, on the other hand, between 2 males, as this species have this rock dwelling behavior, it's kind of easy to make them behave because it's only we just private them from eating like 1 hour before the experiments, and then they're good to go. And then we used the time basking as a measure. So what we have is like this pretty much all the time. So our preliminary results, the color polymorphism in relation to intersexual selection context in the visual signaling experiments, we have no difference between the morphs. In relation to chemical signaling, we also had no difference between the morphs. But what we have here what we have is the male pairs. Each of these is a male pair and a standardized observation. So towards everything that is positive is towards the black male, everything that is negative is towards the yellow male. What we have here is that some individuals are, for some reason, really, really chosen, but we don't know why, and this is not yet, but this is not related to the morphs. So then with the intrasexual selection context, the experiments, we had no outcome difference between morphs as well. So our ongoing work and our future steps, we are intending to link the morphs to a performance measure. So we already ran all the lizards through sprint speed experiments, and we are still analyzing the data. And we intend to do a bite force measure as well to link as a performance measure. Although I'm not interested in the patch itself, but all the traits that can come with it. So between these two groups, we are doing spectrophotometry. Actually, we collected the data for all the lizards already, but we are analyzing the data. We intend to assess parasite load too. We already collected blood smears from the lizards to assess the parasite load. We are analyzing it too. And immunocompetence. So this we made through our PHA experiments, which is the phytoegomycinine, which we inject in the lizards, and then we can measure the swelling of the part, as this is an example of using it. So the experiment is done, but we don't have it analyzed yet. And another thing that I intend to do is to link with environmental traits, let's say this way. So for every single lizard that we caught, we have this the identity, the individual GPS location, and I intend to map them in distribution within the population. We also took, I want to make a vegetation coverage index, where for every point of it, I took we have a photograph of the vegetation coverage, so we can make a analysis in how much the basking place is exposed or not. And thermal stratification. So each individual, we collected the body temperature at site at first cut and the ground temperature as first cut. But these all together, we are still thinking on how to use it. We thought which kind of questions we want to ask to answer, but how to do it is we are still thinking about it. So, well, I would like to thank you very much for your presence here, both the university and the labs, and both my advisor and co-advisor, and these, besides the co-authors, all these people that keep helping me during the process. And also, if you want to discuss anything about this work, or I'll be presenting my other work as in the poster today. So if you want to discuss anything about this, or about the evolution in bioacoustic patterns that I made, I'm happy to do so. So thank you very much.